Okay, so that's how. So medical marvel for the future, and that's something that's really on its way. We're very, very far advanced in that field. Genetically modified plants, another thing. These are on the market. You've probably seen and heard of lots of people debating these. Over time, we have modified and bred plants to create lots and lots and lots of new types of crops. Just about everything that's out there that you eat was not what it looked like originally. Well, there's we only eat female corn? I see what you're saying. Right. You've got different parts. Corn is it produces the male part of the plant is in a different place than the female part of the plant. Oh, the male's at the top. And right. All over. Okay, so it's what's it called? Perfect. That's what it means. It's the perfect plant. Perfect. So we've created these new types of crops. You guys have seen tomatoes, and you know they didn't look that huge thing before. If you've ever gone walking in like some wild-looking woods, you don't see giant fruits hanging from trees. Ooh, except the pawpaw tree. Have you ever heard of the pawpaw tree? It might be called the horse apple. I've decided I'm getting a pawpaw tree. I'd learned about these. They're native to Virginia. You don't find them often, but they make these large fruits that are really sweet that only last for like 2 days before they rot. So you can't sell them, but I could have them at my house. I think they're green. What? No, not those. Those things you just throw at each other. That's what those are for. Crab apples. We can modify plants not just using the old way of using ultraviolet radiation from the sun, but through lots of different ways. Agrobacteria transformation, biolistics, microinjection, and protoplasts. Pretty much you take those totipotent stem cells and you trick them into doing what you want. For Agrobacterium, Agrobacterium is a natural bacterial genetic engineer. It is way more efficient than we are. We've been trying to perfect genetic modification. These guys did it a long time ago. What they do in their natural state is they induce tumor formation in plants. They effectively cause plants to have cancer. Because these tumors are going to provide a house for the bacteria. So what we did was we looked at that and said, well, in order for them to do this, they needed to trick the plant into expressing bacterial genes. Which means that if we can modify the bacteria, if we can change the genes in the bacteria from build a house to glow green and infect the plant, then we can make the plant glow green. So what you do is you change the bacteria. If you've changed the bacteria, you genetically modified it, you've added a new gene in, you then get the bacteria to infect the plant. The bacteria inserts your gene into the plant. Now you have a plant cell that might glow green. You make that plant cell grow. You give it some, we haven't talked about like auxins and cytokinins. That's next semester for you guys. You give it hormones. You guys know what hormones are, right? You've experienced them. Hormones will tell the plant, grow roots, grow shoots, until you get a plantlet that eventually becomes the transgenic plant. Every single cell in this plant, produced by that one cell, is going to express your gene. Fully, yeah.
Do you think they'll ever change pineapples to pink pineapples?
To pink pineapples? Why would they do that? Do you want a pink pineapple? Is there a market for a pink pineapple?
I would like to. There's
So pink pineapple, you could just add a dye.
But there are no pink ones.
Ooh. You could engage in, honestly, I don't know about making a pink pineapple. If there was a market, somebody would make it.
I know Texas A&M University is really good about making multicolored vegetables and fruits. They probably have already made.
There you go.
Pineapple. I know they make maroon carrots, and they made some flowers that only come in orange and white. They made those purple also.
Okay.
So they're all about that.
Another way to modify a cell is through what's called biolistics. And it sounds sort of like ballistics, right? Because that's what it is. You load up, you coat little balls with DNA, the DNA you're interested in sticking into the organism. You load them into what we call a gene gun. Not kidding, that's what it's called. And then you fire it at your cells. It does. It's literally, it's called a shotgun approach because you are blowing holes in this genome and hoping that some of your DNA sticks. It doesn't work often, but if you're dealing with 100,000, a million cells, and you hit them all, it works. You get one or two that work, and then you've got a transgenic cell, which will then you can then cause using hormones to grow shoots, grow roots, and then you have a whole plant. This also works on other animals and such, but again, the success rate is so low, it's really not worth doing. It's expensive. Microinjection is more precise. It sounds just like what it is. You take a needle that's got your you got your DNA in solution, you get it through the cell membrane, and you inject it directly into the nucleus. Sometimes it works, sometimes it doesn't. I'll reject you. And you could use these naked cells. This is where the cell wall's removed, which makes it really, really easy to just trick something in through the plasma membrane. Protoplast.
There are many uses for transgenic plants. It could be to increase the plant's protection from insects. It could be used for increasing plant quality. It could even be used to create new products. New genes, for instance, could be added to stop viruses from attacking plants or bacteria. It could prevent fungus from invading or prevent insects from eating them. Heck, it could even stop plants from dying in extreme drought or high heat or too much water. What companies have made a lot of money on recently is introducing resistance to herbicides so that you could spray a crop filled with weeds and only the weeds would die, not the other plants that you planted. You could change the composition of a plant. You can increase the amount of nutrients present. This may alter the length it could be stored. It could allow people to eat it weeks or months later. It's a natural preservative. Or you could create new products. Antibodies, pharmaceuticals, vaccines, biodegradable plastics. If you can think of it, we can make it using biotech. Now, once you've got a single genetically modified plant, it can be cloned. That's making a Xerox copy again and again and again of the same plant. It makes genetically identical organisms. That first plant you make is very, very expensive. That first plant you make is hundreds of man-hours, thousands of dollars in chemicals just to produce one plant. But once you have that one plant, you can clone it again and again and again and again. And now you have a product.